Welcome to G Fun Facts Online. Today, we're diving deep into a technology that's on the verge of revolutionizing medicine as we know it, a tool that promises to rewrite the very code of life. We're talking about CRISPR. You've probably heard of it, but the story is moving faster than anyone imagined. We're now entering the era of CRISPR 3.0, a new generation of gene editing that offers unprecedented precision and is poised to tackle a vast array of genetic diseases. To understand where we're going, we first have to understand where we've been. Let's do a quick recap of the original revolution, what we can call CRISPR 1.0. This system, CRISPR-Cas9, was adapted from a defense mechanism found in bacteria. Think of it as a microscopic immune system. Scientists harnessed this to create a two-part gene editing tool. You have the Cas9 protein, which acts like a pair of molecular scissors that can cut DNA, and you have a guide, RNA, a small programmable molecule that tells the scissors exactly where to cut. When you put them in a cell, the guide RNA leads Cas9 to a specific spot in the genome, and snip, it makes a cut across both strands of the DNA, what's known as a double-strand break. This targeted cut is what makes it so powerful. The cell then rushes to repair the damage using one of two main pathways. The first is called non-homologous end joining, or NHEJ. This is the cell's fast and messy emergency response. It often inserts or deletes a few DNA letters at the cut site, which is great for researchers who want to knock out a gene to see what it does. The second pathway is homology-directed repair, or HDR. This one is much more precise. If you provide a correct DNA template, the cell can use it to repair the break flawlessly, allowing scientists to correct a faulty gene or insert new genetic information. The simplicity and power of CRISPR-Cas9 sparked a research explosion and led to incredible breakthroughs, including the first FDA-approved CRISPR therapy, Casgevy, for sickle cell disease. But, despite its transformative impact, CRISPR 1.0 isn't perfect. The very thing that makes it powerful, the double-strand break, is also its biggest weakness. Think about it. Cutting both strands of the DNA helix is a drastic action for a cell. While the messy NHEJ repair is fine in a lab dish, it's far too unpredictable for clinical use, where it could cause harmful mutations. And the precise HDR pathway, the one you'd want for therapies, is incredibly inefficient in most cells in our body, especially non-dividing cells, like neurons and muscle cells. Worse still, these double-strand breaks can sometimes lead to major, catastrophic genomic rearrangements. We're talking about large chunks of DNA being deleted, or whole chromosome arms getting shuffled around, which can trigger diseases like cancer. On top of that, there's the problem of off-target effects. The guide RNA, while specific, can sometimes make a mistake and lead the Cas9 scissors to a similar-looking sequence elsewhere in the genome, causing unintended cuts. These limitations made it clear that to unlock the full therapeutic potential of gene editing, we needed a safer, more precise approach, one that could make changes without the collateral damage of a double-strand break. And that brings us to CRISPR 2.0. In 2016, researchers led by David Liu at the Broad Institute unveiled a technology called base editing. It's been brilliantly described as a pencil and eraser for the genome. Instead of cutting the DNA like scissors, base editing chemically rewrites a single letter of the DNA code without ever breaking the DNA backbone. Here's how it works. Base editors are fusion proteins. They take a modified Cas9 protein that has been disarmed. It can still be guided to a specific DNA location, but it can no longer make that dangerous double-strand cut. This disabled Cas9 is then attached to an enzyme called a deaminase. So the guide RNA takes the whole complex to the target, and instead of cutting, the deaminase enzyme gets to work. It chemically converts one DNA base into another. There are two main types. Cytosine base editors, or CBEs, convert a cytosine, C, into a base that the cell reads as a thymine, T. This effectively changes a CG base pair to a TA base pair. Then you have adenine base editors, or ABEs, which convert an adenine, A, into a base the cell treats as a guanine, G. This changes an A-T pair to a GC pair. By directly rewriting single DNA letters, base editing bypasses the need for double-strand breaks and the cell's unpredictable repair pathways. This makes it far more precise and safer for correcting point mutations, the single-letter typos in our DNA that are responsible for thousands of genetic diseases. Base editing has already shown huge promise. In mouse models, it's been used to correct the single mutation that causes progeria, a devastating premature aging disease, significantly extending the animal's lifespans. But, like any technology, it has its own limits. It can only perform those two types of conversions, C to T and A to G. And sometimes, the deaminase enzyme gets a little overzealous and edits nearby bystander bases, causing unintended changes. This pushed scientists to ask, can we do even better? Can we get more versatile? The answer came in 2019, again from David Lia's lab, with a technology that truly earns the title of CRISPR 3.0 prime editing. If base editing is a pencil, prime editing is like a word processor's search and replace function. It's an even more versatile and precise tool that can make all 12 possible single-base substitutions, plus it can make small insertions and deletions of DNA letters. And crucially, it does all of this, again, without making a double-strand break. Prime editors also use a modified Cas9 that only nicks one strand of the DNA, which is easily repaired. But instead of a deaminase, this Cas9 is fused to a different enzyme, a reverse transcriptase. This is an enzyme that can write new DNA based on an RNA template. The real genius of prime editing is in its guide RNA, called a pegRNA. This isn't just a guide. It's a guide and a template all in one. It contains the sequence to find the target DNA, but it also carries an extension with the RNA template for the desired new edited DNA sequence. The process is a masterpiece of molecular engineering. First, the search. The pegRNA guides the prime editor to the right spot. Then, the nick. The Cas9 nicks one strand of the DNA. Next comes the magic. The newly nicked DNA strand binds to the pegRNA, and the reverse transcriptase enzyme uses the RNA template on the pegRNA to synthesize a new stretch of DNA containing the edit. Finally, replace and repair. The cell's own machinery recognizes the newly synthesized DNA flap and integrates it, replacing the original sequence. The old, unedited strand is then removed and replaced with a new one that perfectly matches the edited strand. The versatility is stunning. Because the edit is encoded in the pegRNA, you can, in theory, correct a much wider range of mutations than with base editing. This includes the small deletions that cause diseases like cystic fibrosis and Tay-Sachs, or the single base change behind sickle cell anemia. Prime editing also appears to be incredibly safe, with very low off-target effects. This is because it requires 3 separate molecular handshakes to happen before an edit is made, making it highly unlikely to occur at the wrong place. Of course, it's a newer, more complex technology. Its efficiency can sometimes be lower than base editing, and its larger size can make it harder to deliver into cells. But researchers are rapidly making improvements. And the CRISPR 3.0 toolkit doesn't stop there. Other next-generation technologies are emerging. For example, CRISPR-associated transposase, or CASTs, are being developed to insert very large pieces of DNA into the genome without double-strand breaks. This could be a game-changer for diseases like cystic fibrosis, where you need to insert a large, healthy copy of a missing gene. Other systems, using a protein called Cas13, can edit RNA instead of DNA. RNA is the temporary messenger molecule that carries instructions from our genes. Editing RNA allows for temporary changes to gene expression, a potentially safer approach where a permanent genomic change isn't needed. There is also epigenome editing. This uses a dead Cas9 to carry enzymes that add or remove chemical tags on our DNA. These tags control which genes are turned on or off. So without altering the DNA sequence at all, we can correct diseases caused by faulty gene regulation. The promise of CRISPR 3.0 isn't just theoretical, it's rapidly moving toward the clinic. Base editing has been used to cure sickle cell disease in mice and correct the Huntington's disease mutation in patient cells. The first human trials for base editing therapies are already underway. Prime editing is close behind, having successfully corrected mutations for various genetic diseases in human cells and animal models, with clinical trials expected in the near future. As these technologies become more powerful, the ethical questions become more urgent. The increased safety of CRISPR 3.0 might make the prospect of germline editing, making heritable changes to the human genome, more tempting. The scientific community is largely against this for reproductive purposes right now, but the conversation is far from over. And then there's the issue of access. The first CRISPR therapies are expected to cost millions of dollars per patient. How do we ensure that these life-changing treatments are available to everyone who needs them, not just the wealthy? We are standing at the dawn of a new era of precision medicine, moving from just treating symptoms to truly correcting the root cause of genetic disease. CRISPR 3.0, with tools like base and prime editing, gives us the ability to fix genetic defects with an accuracy we could only dream of a decade ago. The road ahead is long, but the potential is immense. The ability to rewrite the code of life is here. And with it comes the profound responsibility to wield this power wisely for the benefit of all humanity. Thanks for listening to Fun Facts Online.